Hello Bio 130 and welcome to this video lesson on the main organs of the digestive system. Now just to get a little bit further into the nucleus, mitochondria, and ribosomes, which were discussed in the last lesson. Some of this material was already discussed, but just to give a little bit further, where the nucleus is what houses the DNA of your body, of your cells. This is the general site where many viral diseases will attack, including the HIV, a retrovirus that attacks by inserting its genetic code into your own. There is also, within the mitochondria, the energy factory of the cell. The mitochondria consists of many folds, which give it lots of surface area to help produce more and more energy molecules. Now, those folds on the inside of a mitochondria are susceptible to attacks from oxidants and free radicals, which essentially smooth out the surface, the inner surface of a mitochondria, removing much of the surface area and diminishing its ability to produce energy molecules, which is ATP. And then for ribosomes, which are the protein manufacturers, there are a variety of diseases called ribosomopathy, which could be any diseases afflicting ribosomal construction. Now, for this class, we're going to get into diseases. For some body systems, we will get into medical technology. So that was just an introduction into some diseases that affect on the cellular scale. But we're now going to get into the digestive system. And the digestive system consists of two main types of organs. The first type are the hollow organs, which are essentially the organs that food will travel through. But there are also going to be solid organs, which will act as accessories to the hollow organs of the digestive system. And the purpose of the digestive system is to fulfill the functions of ingestion, digestion, and absorption, taking in food, breaking apart that food into smaller and smaller components and molecules that can then be absorbed later into the body. This is where carbohydrates will be broken down into simple sugars, proteins will be broken down into amino acids, fats will be broken down into smaller fatty acids and glycerol, and this will also be where you will see the absorption of vitamins, minerals, and water. The digestive system for what is called the GI tract, digestive tract, alimentary canal, these are essentially the connected hollow organs, which go more or less in a continuous chain. And the hollow organs would be the mouth, esophagus, stomach, and then the small and large intestine. When it comes to the digestive system, there are two types of digestion to think about, which would be mechanical and chemical. And this will also be important to consider for your experiment this week in the digestive system. When it comes to the mouth, you will see both types of digestion occurring. Mechanical digestion, which is done by the grinding and chewing of your food with your teeth, and then chemical digestion, which is through the release of salivary amylase in your saliva. Now, where mechanical digestion in your mouth will help break apart food into smaller and smaller bites that are easier to swallow, the chemical digestion that occurs with salivary amylase essentially takes starch, which is a molecule consisting of long chains of glucose sugars connected together, and breaks them down into maltose, which is a disaccharide consisting of two glucose molecules connected together, as opposed to 10, 20, or many, many more, which is what starch is. Now, there is a term for the material that you swallow, that material which is partially digested starch and saliva and food mixed together. That will be called a bolus. The next organ after the mouth is going to be the esophagus, which is essentially the long tube that connects the mouth to the stomach. There is no digestion that really occurs in the esophagus, but this will be the start of a process called peristalsis. Peristalsis will continue through the stomach and the small intestines. This is the body's way of, through involuntary wave-like motions, moving material throughout the rest of the digestive system. And this is done through muscles throughout the body and the digestive system, constricting and relaxing. The key emphasis is that in peristalsis, it's an involuntary movement, so you do not need to consciously think about pushing through food through your esophagus, through your stomach, through your intestines, which is definitely a big bonus. Now, the stomach will be the next major hollow organ. Like in the mouth, you will see both mechanical digestion and chemical digestion occurring in the stomach. For the stomach, the upper muscles will relax, which allows the bolus to leave the esophagus and enter into the stomach. And then the lower muscles will contract and help mix the bolus with gastric juices. And this new combination of material is called chyme. Now, as your stomach is grinding and churning that food, helping break it down into smaller and smaller physical chunks, there is also chemical digestion occurring thanks to your gastric juices. Now, those juices are essentially stomach acid, which is composed of hydrochloric acid. This helps unfold protein chains so that they're much easier for enzymes to get into, whereas proteins might be difficult in their original folded, clustered state. Now, thanks to the stomach acid helping to unfold and unravel protein chains, pepsin, the enzyme that's in the stomach, can then get to work. And what pepsin does is it helps break down large proteins into smaller and smaller fragments, which can then be broken down even further in the intestines. Similar to the esophagus, peristalsis is what helps continuously push chyme from the stomach further down into the small intestine. Now, digestion will continue in the small intestine, and in the small intestine, this is where major absorption of those components, carbohydrates, fats, proteins that were broken down further and further. The small intestine is where absorption of those materials truly begins. And the small intestine can be broken up into 3 regions, which aren't distinguishable if you were to actually physically look at them, but they are distinguishable in the kinds of activity that occurs in those portions of the small intestine. The three components of the small intestine, or the 3 regions of the small intestine, are the duodenum, jejunum, and ileum. As chyme enters the small intestine, the first part of the small intestine is the duodenum. This is actually where the accessory organs start adding assistance to the digestion of material. The accessory organs are going to be the pancreas, the liver, and the gallbladder. Food does not travel through these accessory organs, but the accessory organs can pump different digestive material into the duodenum to help break apart what wasn't completely broken down in the stomach. What the pancreas adds to the duodenum are quite a few very important things. As the as the chyme is entering the small intestine, it's going to be very acidic thanks to the stomach acids. So the pancreas will actually release sodium bicarbonate, a base, to help neutralize the stomach acid. The pancreas will also release trypsin and chymotrypsin, which further breaks down proteins into smaller and smaller peptides and amino acids. The smaller you can break down different food material, the more easier it will be to absorb that material. So that's key. The pancreas will also release pancreatic amylase, which, similar to the mouth, which had salivary amylase, pancreatic amylase further breaks down starch. Lipase will also be released, which helps break down lipids, aka fat molecules. And then there are also nuclease and nucleosidases, which break down nucleic acids, which are found in DNA, ribonucleic DNA of the food that you ingest, that you swallow. Now that is a bunch of things that the pancreas does to help act as an accessory organ to the hollow organs of the digestive system. The liver and the gallbladder are very important in aiding fat digestion. The liver will produce a substance called bile and then release it into the duodenum. Now, bile acts as an emulsifier. This basically means that it helps keep fat molecules still and keeps their long chains from essentially clumping together. Since fat molecules consist of long chains that like to stick together, that makes it hard for enzymes to, on their own, squeeze into fat molecules and slowly digest and break them apart into smaller pieces. So because the bile produced by the liver helps emulsify or keep still and keep spread out fat molecules, digestive enzymes can get in there and help break apart those fat molecules into smaller components and fragments that will be more easier to absorb. Now, as food quickly enters, or as food enters directly from the stomach to the duodenum, the liver, there might not be bile already produced by the liver to help begin fat digestion. So the gallbladder will actually act as a reserve of bile. That way, when food enters the duodenum, there's already bile stored in the gallbladder that can then be released into the duodenum, giving the liver time to produce more bile if it's needed, or to produce reserve bile, which will be stored in the gallbladder. Now then, the duodenum there will also be different enzymes that will be produced by the duodenum or inside the duodenum. And most of these enzymes actually help break down sugar or carbohydrates. One of them is maltase, which helps break down maltose into glucose, individual glucose molecules, individual glucose sugars. There is also sucrase, which is an enzyme that breaks down sucrose into individual fructose and glucose sugars. Lactase will also be made, which helps break down lactose into individual molecules of galactose sugar and glucose sugar. All these individual sugars are called monosaccharides. Now, there is also peptidase, which is produced. And what this does is it further breaks down protein fragments into small peptides and amino acids, which are much more readily absorbed into the body, or will be easier to absorb into the body later on. Now, key about the duodenum is that this is the point where major nutritional absorption begins. Whereas in the mouth, there was a little bit of digestion. In the stomach, there is just digestion of protein material. The duodenum helps find all the pieces to help further digest everything else, and then start absorbing carbohydrates and fats, essentially the components and fragments that were broken apart. This is where absorption starts happening. Now, the other two main components of the small intestine are the jejunum and the ileum. And this is where digestion of macromolecules, of large molecules, carbohydrates, fats, proteins, digestion of these large molecules finishes in the jejunum. And the main purpose of the jejunum is to actually absorb nutrients, where the duodenum's main purpose was to break down, was to do the majority work of digesting food material. The jejunum's main purpose is to absorb, and just absorb more and more. Within the jejunum, carbohydrate fragments, protein fragments, fat fragments will be absorbed. The ileum serves as essentially the main site of absorption for what's left. These can be the fat-soluble vitamins like vitamin A, vitamin E, vitamin K, as well as vitamin B12, as well as leftover bile that wasn't degraded in the earlier parts of the small intestine. That will be absorbed as well. Now, we will get into the last major organ, as well as a few diseases of the digestive system. The last major organ, which is the large intestine, but that will be in the next video lesson. Something fascinating to look at is this chart, which essentially shows a lot of the enzymes that are at play throughout the parts of the digestive system that were discussed. Something to note is to look at how different enzymes would be useful in just breaking down carbohydrates. Some will be useful at only breaking down proteins. Something to look at is how salivary amylase, which is active in the mouth, occurs at a neutral pH. Naturally, you would not want your mouth to be too acidic or too basic, because that can cause damage. Whereas the pancreatic amylase, which is active in the small intestine, that is working at a basic pH. And naturally, pepsin, which is in the stomach, is active at an acidic pH. Just something to note is that enzymes, certain conditions, certain parameters have to be occurring in order for those enzymes to actually function. Those parameters can include certain temperatures. In this case, the key component is the pH level. Is the pH acidic, basic? Is it neutral? Notice how in the stomach it's acidic, whereas in the small intestine you're looking at basic conditions. That's key. If those enzymes were occurring in different parts of the digestive system, they would not actually be functional nor effective at breaking down food material, because they are limited to a certain pH range. And that's kind of cool. That's essentially throwing in chemistry into this basic anatomy course. For your discussion, go to the discussion section, and each of you will be responsible to make a thread to discuss in at least a few sentences the material discussed in this video lesson. Now, you can discuss it in a variety of ways. You could summarize the material that you learned, key points that stuck out to you, opinions of what was discussed. For example, perhaps you didn't know this, or perhaps you were familiar with this certain material. Essentially discuss the lesson material. Well, yeah. Discuss it in a way that makes sense to you. You will also be responsible to reply to another of your classmates' thread, to a thread produced by another one of your classmates. Now, your reply could include thoughts on their discussion thoughts on their discussion, like opinions on what they got out of the discussion lesson or what stuck out to them. You could also include or just limit your reply to relating different organs in the digestive system through anatomical directions and terminology to help further hone those important terms. So, within the 21.5-minute mark, this concludes our first lesson into the digestive system. And as always, it is a pleasure to have you guys as students.